Extraction techniques for food analysis. Let's look at some of the key methods and applications. Food items in general range from fruits and vegetables to meat, dairy, coffee, nuts and flours. Foods are composed of mixtures of sugars, proteins, vitamins, lipids, fiber, polyphenols, antioxidants and organic compounds. These are usually present in small quantities comprising of a diverse range of chemical compounds. Due to the diverse characteristics and immense value of food components, they have to be processed and preserved accordingly to retain the nutritive ingredients, aroma and flavor. The necessity to consume healthy and diet-rich foods has led to a deeper investigation into the inner components of food products. Looking at the need of extraction techniques in food analysis, in the realm of food analysis, extraction techniques play a crucial role. They help in isolating flavors, aromas, and nutrients from food matrices. The real challenge lies in utilizing a suitable extraction step or process for the isolation of compounds from the source. These techniques should be cost-effective and perform in an energy-efficient manner without disturbing the structural integrity of the sample. In food processing, extraction is defined as the transfer of one or more components of a biological feed from its source material into a fluid phase, followed by a separation of the fluid phase and recovery of the components from the fluid. The feed is usually of plant origin, but the principles of extraction remain the same if the material is animal or plant in origin. Let's now start with understanding a few basic concepts. A component that is desired to be removed from the feed, that is the sample, through extraction is called as a solute. The phase that is mixed with the feed to remove the solute is called as the solvent. After the solvent has been mixed with the feed and the solute has been transferred from the feed phase into the solvent phase is called as an extract. So we have three important components to be understood: the solute, the solvent and the extract. In food processing, the feed is usually solid, semi-solid or gel-like, whereas much of the science of extraction is based on liquid foods. Types of extraction techniques. There are two types of techniques. One, conventional techniques. The other one is the modern or advanced techniques. Conventional techniques consume a lot of time. They use huge amounts of solvent and energy, thereby leading to high costs. There are three types of conventional techniques: solvent extraction, Soxhlet extraction, liquid-liquid extraction. Among the advanced and modern techniques are supercritical fluid extraction, ultrasound assisted extraction, microwave assisted extraction. These advanced techniques are energy efficient, consume less or no solvent. They are safe as well as they produce a high quality extract. However, research has also shown that the hybrid technologies could be used, and the importance is being investigated as they combine two different processes aiming to improve the effectiveness of the process. Let's look at each one of the techniques. Conventional techniques, solvent extraction. This is a process in which the compounds are separated based on their relative solubilities. So, solvent extraction is defined as the technique of removing one constituent from a solid by means of a liquid solvent. It is also referred to as leaching. The principle of solvent extraction is that a solvent selectively dissolves or separates the desired component from the mixture. The second one is Soxhlet extraction. This is a laboratory technique used to extract compounds from solid materials. It works on the principle of a continuous extraction using a refluxing solvent. This is a well-established technique that is efficient and reproducible and is often used in oil and food industries. The applications include extraction of lipids from food samples for nutritional labeling and analysis, and it is also used in pesticide residue analysis. The advantages are it has a small amount of solvent is reused, making it cost effective, that is, with regard to the solvent efficiency. Secondly, it is a continuous operation and hence it can extract compounds over a long period of time. The third conventional technique is liquid-liquid extraction. Liquid-liquid extraction, or also known as LLE, is a method for separating compounds or metal complexes based on their relative solubilities in two different immiscible liquids. This technique is also referred to as solvent extraction or partitioning, and the principle partitioning between two immiscible liquids. In liquid-liquid extraction, it involves adding an aqueous solution containing the solute to a separatory funnel and then adding the non-water soluble organic solvent. The organic compound partitions into the organic phase because it's more soluble in that phase than the aqueous phase. The advantage is that it is straightforward, efficient, and can be used with low sample volumes. It is also applicable to heat-sensitive compounds. Looking at the applications, it is used in food processing industry to concentrate valuable components of natural raw material as well as to remove undesirable or potentially harmful components. The main disadvantage of LLE can be challenging when extracting multiple analytes with different properties. Finding the right solvent can also be difficult. Intense mixing can lead to emulsion formation, which can slow down the separation process. Coming to the advanced extraction techniques, also referred to as emerging technologies. The first is supercritical fluid extraction, for which a schematic diagram is shown here. This is a method for isolating a specific compound from a mixture of chemicals using pressurized fluids as solvents. It works on the principle of using pressurized fluids to separate a desired compound from a mixture of chemicals. How does it actually work? The raw material is placed in an extractor vessel, which is pressurized with supercritical fluid. The supercritical fluid is a fluid that has been forced to a pressure and temperature above its critical point, which causes the liquid and gas phases to become indistinguishable. The fluid dissolves the compounds in the raw material, which are then transported to separators. Looking at the challenges involved, it is expensive, it requires specialized equipment, and can result in low recoveries or high levels of impurities. Generally, the applications include decaffeinating green coffee beans and extracting oils from hops. The next advanced technique is ultrasound assisted extraction, also referred to as UAE. This is a technique that uses ultrasonic waves to extract compounds from plant matrices. UAE works by using ultrasonic waves to create acoustic cavitation, which produces shock waves, microjets, and high shear force. These forces disrupt the cell walls, reduce the particle size, and improves the contact between solvents and target compounds. Normally, UAE can be used to extract natural pigments from food byproducts. It can also be used in the production of herbal extracts with anti-cancer, antimicrobial, and anti-diabetic properties. UAE has been referred to as a clean technology. This involves preparation of samples with high purity. Advantages include it is a fast and efficient process. The environmental impact, it requires less solvent. It is a low-cost technique, simplicity, and produces high yield of the product. The next is microwave assisted extraction, which is MAE. Microwave assisted extraction is a technique that uses microwave energy to heat solvents in contact with a sample in order to extract analytes from the sample. It enhances the evaporation of raw material, residual water, and eventually breaks the plant cells to promote the extraction through internal diffusion. MAE is popular and cost-effective method for extracting compounds from natural products. This is different from those of conventional methods because the extraction occurs as a result of changes in the cell structure caused by electromagnetic waves. Advantages of MAE is faster extraction, uses less solvent, higher extraction rate, lower cost, and improved reproducibility. MAE can extract multiple samples simultaneously. So, having looked at the different kinds of techniques, this is a summary of the supercritical fluid extraction and advanced technology, which is an efficient method for isolating and purifying active ingredients from natural and synthetic raw materials. Ultrasound assisted extraction, again a modern technique. It is useful for extraction of vitamins, antioxidant compounds, flavonoids, phenols, polysaccharides, alkaloids, and natural compounds. MAE or microwave assisted extraction is a technique that uses microwave energy to heat a solvent and extract the analyte from a matrix. And the most common oxalate extraction is a safe method for extracting fats and pesticides from food samples.